The specifics vary from study to study, but many show results just as surprising as those that McCay discovered so many years ago. The exact manner by which calories are reduced changes across studies, but the animal's calorie intakes are generally reduced by as much as half their normal levels. Depending on the species, the most promising results have shown lifespan extensions between 50 and 300%. If these results hold water, then they could have important implications for our own longevity. However, these studies are not without controversy. Many scientists are concerned that calorie restriction will not benefit humans, or that it could even result in long-lasting harm. Is calorie restriction a silver bullet against aging, or something that should be kept in the laboratory? The exact mechanisms by which calorie restriction extends lifespan are still being studied, but two prominent theories offer some insight. At first glance, calorie restriction is a counterintuitive way to approach longevity. Here, we use this term to refer to up to a 50% reduction of calories from a normal diet, not simply a lack of overeating. Consuming so much less food than a normal diet seems like it should reduce your lifespan, not extend it. To better understand where this idea comes from, it's necessary to first understand how aging, the process by which our bodies function less well over time. There are two classic theories that could potentially shed some insight into this process: the rate of living theory and the free radical theory. The rate of living theory arose from the observation that larger animal species tend to have longer lifespans than small ones. One way to explain this observation is through the concept of metabolic rate, which refers to how quickly an animal expends energy for the body's day-to-day functioning. This includes energy needed for processes like breathing, maintaining body temperature, and circulating blood. Since larger animals have also been observed to have lower metabolic rates, this theory suggests that slower metabolic rates, i.e., slower rates of living, are associated with longer lifespans. The free radical theory provides a potential explanation for the rate of living theory. Under this theory, aging is a result of the cells in the body accumulating damage over time. This damage comes from so-called free radicals, which are highly reactive atoms or molecules naturally produced by our bodies. They can damage proteins, DNA, and fatty tissue, which in turn is theorized to eventually cause age-related diseases, such as heart conditions, neurodegenerative disorders, or cancers. Because free radicals are created by our metabolic processes, slower metabolisms could decrease the rate of free radical production and subsequently extend lifespans. Calorie restriction enters this equation because it is known that drastically reducing food intake will reduce metabolic rate. If less food is being consumed, then there is less food that the body has to process. Moreover, since calorie restriction generally results in weight loss, less energy overall is needed to maintain the reduced body mass. As a result of this reduction of metabolic rate, it is hypothesized that calorie restriction could extend lifespans by decreasing the rate of free radical damage. This idea is further supported by direct evidence that some species produce fewer free radicals under calorie restriction. This suggests that calorie restriction slows down the aging process, allowing the organism to live longer and with less risk of age-related diseases. Does calorie restriction have a place outside of the laboratory? One of the largest critiques of calorie restriction studies is how the control group is treated, which is a common issue across the wide range of species tested. While the experimental group animals are placed on a highly restrictive diet, many studies allow the control group to eat as much as they want. The control group often ends up consuming much more than they normally would in nature, which can lead to a number of weight-related diseases and poorer health overall. Even putting the concerns with these studies aside, many scientists are skeptical of applying results from other species to humans. It is much more challenging to carry out calorie restriction studies in humans, since we simply cannot, and should not, exert the same degree of control over human subjects as we might for rats. As a result, there have been very few studies in humans. The most prominent such human study was the Calorie trial, a randomized clinical trial in which healthy people were divided into a calorie reduction group and a control group. Because the trial only lasted for 2 years, the effects on lifespan could not be directly measured, and the goal was to instead investigate the effects on typical markers of age-related disease risk. The calorie restriction group was told to reduce intake by 25%, but in practice, they only reduced their intake by about 12% on average. Nevertheless, there were still significant health benefits observed in this group. This included lower cholesterol and blood pressure levels, as well as improved insulin sensitivity index. However, this trial alone is not enough evidence to conclude that calorie restriction should enter standard medical practice. Much more research is needed, but human studies face many limitations. Calorie restriction is a challenging protocol to follow and likely to be met with low compliance. Moreover, asking individuals at a healthy weight to dramatically reduce calories, as animal studies have been doing, poses ethical concerns. Clinical research on eating disorders has amply shown the negative consequences of excessive restriction and malnutrition, so trials would have to operate very carefully to avoid risking the safety of their participants.